Is it recording? Good. Okay.
All right, well, let's take a look. Once again, we're looking at the regulation or level of transcription. We mentioned in eukaryotes, we have ourselves this promoter sequence with our Tata box. Almost all these things have Tata boxes. And here's our promoter. And then about 30 base pairs further on down, you've got your transcription start site, and a couple more nucleotides, and you have your first exon. And of course, we mentioned before, for those of you who weren't here Thursday because of that weather, fortunately it wasn't as bad as they had predicted, but the key thing in the promoter, what assembles together is not just the RNA polymerase, it's the polymerase and a whole slew of other proteins that we call general or basal transcription factors to form this huge thing called the basal complex. The cornerstone of, and the first thing that binds when we assemble basal complex together is going to be this protein called a Tata binding protein, or TBP. And that's the foundation of the basal complex. Other proteins assemble onto it. You have a number of these basal transcription factors that includes things like TF2D, or rather, and TF2F, TF2H, those types of things. They assemble together, grab onto the polymerase. We went through this already. Grab onto the polymerase, position the polymerase just right, and there are a couple other transcription factors, basal transcription factors, that do things like, one of them is a helicase. It unwinds the DNA. You know, DNA is a double helix, a double spiral strand, and if you just kept on pushing through it, you would eventually wind up the DNA in front really tight, what they call supercoiling, and then it gets really hard, and it actually finally gets to a point where it's impossible for polymerase to go through. So we have these, one of these transcription factors is a helicase enzyme. It cuts one strand of DNA, allows it to kind of unravel, and then glues it back together again. And then we have another one of the basal complex proteins is going to be a kinase that phosphorylates the tail end of one of the subunits of polymerase II. And ordinarily, polymerase II is inactive unless it is specifically phosphorylated. So we have a whole bunch of these proteins that join together to form the basal complex. And the basal complex is the minimum requirement for transcription. If you don't have the basal complex fully assembled together, transcription simply won't take place. And what holds it together are going to be a combination of some of the proteins bind directly to DNA sequences. TBP is a perfectly good example of that. Other proteins bind to some of the proteins in the basal complex to stabilize and hold the whole thing together, and still others do both. So we have a whole bunch of things, protein-to-protein interactions and protein-to-DNA interactions that keep this big, huge thing. And we're talking about molecular weight in the order of probably 500,000 or more. It's a huge assembly of proteins. And we have to hold that in place, keep it stable long enough for transcription to actually start. We assemble it together. It takes a certain amount of time to assemble together. It takes a certain amount of time to actually fire the thing off, to actually start transcription. And that's one of the problems we have here when we look at this. The basal complex is difficult to assemble, and here again, it's held together by fairly weak interactions, whether you're talking about binding to DNA or binding to other proteins. None of that stuff is very, very strong. So the problem is, proteins here again, you see these static pictures of proteins and like, protein structures and stuff in textbooks and papers and things like that. But in reality, proteins are in constant motion. They're jiggling around, they're wiggling, they're tumbling, they're flexing, all kinds of things. So a lot of times those kinds of just random movements is enough to break interactions from one molecule to the next, because these are all weak interactions. So the problem comes in that the basal complex may well fall apart before transcription can happen. It's our Transcriptus interruptus, I guess would be a good name for that. Okay, because the thing assembles, it's got to hold together long enough for the thing to fire off. And a lot of times it falls apart. Now that's one of the big things with eukaryotes. Most of these eukaryotic promoters are what we call weak promoters. In other words, they really don't bind the appropriate proteins all that tightly. So, for the most part, the basal complex is simply not sufficient to get much transcription. Maybe a little bit, but you don't get a huge amount for most genes. There are a few exceptions. Now, by the way, a little hint. One reason that viruses do such a good job of taking over cells that they infect is the viral genes have very powerful promoters. They tend to assemble basal complexes. Of course, they're using host cell machinery to do this. They tend to assemble basal complexes and hold them together fairly nicely. So if you get infected by flu, hopefully don't do that. Don't do that, please. You can't afford that. But anyway, if you get like flu, for instance, those viruses, those genes are going to be transcribed preferentially compared to your genes. So it's good for the virus, it's really bad for you. Okay, so that's one of the things. Most of the time, we're going to have to stabilize the basal complex and hold it together with other proteins. And that's where we're going to get our cell-specific gene regulation. And what that depends on okay, a few key points here. The whole secret to cell-specific regulation of transcription is these other proteins, and there are over 1,000 different kinds in our genome that we could call specific transcription factors. These transcription factors are going to be expressed, they're going to be found in certain cell types but not others. And what they do collectively is they're going to bind to various DNA sequences. Frequently they're called enhancers, more on that in a little bit. They bind to various DNA sequences. And then, one way or another, they're going to interact with the basal complex. Sometimes directly, sometimes through adapter proteins. More on that later. But that's a key thing here. If this interaction stabilizes the basal complex, holds it down more tightly, pins it to the promoter, what's going to happen is transcription is going to go up, because now it's less likely to fall apart before transcription starts. On the other hand, some of these transcription factors will do things like push parts of the basal complex off, and that causes the whole thing to collapse. So that means transcription is inhibited. And this is the key to cell-specific regulation of transcription. This is one of the reasons why we have so many different cell types in our own body, even though we all have the same, all these cells have the same genome, same genetic blueprint. For instance, all cells have the genes that code for the proteins called globins. Those globins are the proteins that make up hemoglobin. But the only cells that actually produce hemoglobin are the precursors of the red blood cells, what we call erythroblasts. Now, what happens in this case? In this case, there is a DNA sequence, and the precursors, the erythroblasts, happen to produce a specific transcription factor that no other cell type does. That transcription factor binds this particular enhancer, and it ramps up the transcription rate by many thousands of fold. So, red blood cell precursors crank out huge amounts of the globin proteins and therefore make huge amounts of hemoglobin. Other cells don't do that. The globin genes simply don't get transcribed. The globin promoter by itself is very weak. So, basal complexes, when they start to assemble, they generally fall apart before anything else happens. So in this case, we have one single specific transcription factor unique to one cell type that leads this gigantic production of hemoglobin. And there are other things that aren't quite as dramatic, but it's the same basic principle. So the key thing here, a lot of development, at least when you look at the level of transcription, we've got a couple other levels to deal with later, but the key thing is cell-specific transcription factors. And they somehow affect the basal complex, whether it's directly or indirectly. So that's the fundamental property of these things. Now, when we looked at prokaryotes, a bacteria, for instance, you generally have that one single DNA sequence we call the operator, and you had an activator protein or repressor protein. And here again, that would either stabilize the polymerase or push the polymerase off. But here, when you look at eukaryotes, you have a whole batteries. Many of our genes have 20 separate sequences that bind very specific transcription factors. So that's pretty cool. That's pretty cool. So let's take a look at that.